Okay. Hi, everyone. My name is Josh Earl. I'm an assistant professor at Drexel University. I work in the Department of Immunology and Microbiology. And I'm going to be talking to you a little bit about using machine learning in predicting clinical provenance in Haemophilus influenza. So Haemophilus influenza is an important bug. It's a bacteria. It's one of the leading causes of otitis media, which is a middle ear infection. The incidence worldwide is about 700 million children, like young children, per year. It costs about $10 million in surgeries per year in the US. And sort of overall costs are around like 4 to 5 billion per year in the US. Some of the repercussions are like, it's one of the most common causes of hearing loss in children. So like more than 300 per 100,000. And this can result in a bunch of different things, deficits in like a child's ability to learn speech and language and perform in school and interact with others. So it's an important pathogen to kind of understand. The other thing that we really have to talk about in order to sort of understand a lot of what's coming next is like the evolution of Haemophilus influenza. So Haemophilus influenza is what's called a naturally competent bacteria, which means that it can take DNA from the exogenous environment that it's in and then actually recombine that DNA into its own genome. And so what this means is that not like traditionally what happens is that we all inherit our genetic material from our parents. So we call this like vertical descent, right? Vertical inheritance of DNA from our ancestors. But Haemophilus itself is a strain of bacteria that can actually like sidestep that and end up taking DNA, if the conditions are right, taking DNA from its environment or from other, bacteria that are nearby it or other organisms and actually recombine that into its own DNA, which changes the genetic structure of the genome in these strains itself. And so some of the examples might be like, an organism is particularly fit to exist in some type of environment, but then it can acquire genetic material which gives it a selective advantage in a different environment. And so that's actually going to be pretty important for us in the upcoming slides, because we're going to be using that information to try to figure out if there's something about the genetics itself that predisposes some strains to live in certain environments over others. So in order to facilitate doing this, our labs and others have actually sequenced, whole genome sequenced 1,618 genomes of Haemophilus influenza. And so, we downloaded probably a quarter of the strains from the internet from PNAS and NCBI. But we have also partnered with several other labs in order to get clinical strains in order to do the whole genome sequencing on them. And this slide just sort of shows you some of the types of filters that we use in order to identify what a good assembly actually was. And so we sequenced actually even more than this, but we only used the best sequences that we could get our hands on. So this is sort of like the meat a little bit, I guess, of like what we're going to be trying to predict. So out of our approximately 1,600 genomes, we have labels. Which is really great for analysis, is that we actually have these metadata, these categories that are assigned to each one of these different genomes. And what this means is that a genome that is labeled as carriage means that it was isolated from a person who did not have symptoms and wasn't showing any sort of disease type. And so these are sort of like in a commensal state. And then we have the ear, eye, invasive, and lung categories, which are all different locations in which a strain was isolated from. So this would be things like conjunctivitis, for example, if it was coming from the eye, middle ear infections, if it was coming from the ear, COPD from the lung. Invasive is like if there was a bacteremia or something that was isolated from the blood, for example. two of the categories that we have is, so, you know, some of the strains we just don't know what their provenance was due to, you know, the lack of labeling. And then there were several that were hard to fit into these like categories that we created, and those are the others. And so we have to sort of ignore those when we're doing this classification strategy. And, you know, something that we're going to talk about a lot is this idea of class imbalance and how that's problematic when it comes to doing machine learning algorithms. And so maybe we should be ignoring some of these other categories in here where we have like fewer strains. So, you know, talking about the fact that Haemophilus influenzae is a naturally competent species. What we wanted to do, like initially, is just be like, okay, well, maybe these organisms are just their overall phylogenetic signal, you know, presupposes that they're going to belong to one of these categories already. In which case, it wouldn't really make any sense to do sort of machine learning, because we could just use the phylogenetic signal and say, oh, okay, you know, these are, you know, the reason that they live in the ear is because like everything in this clade lives in an ear, and you know, the ear strains don't exist in other phylogenetic clades. But that isn't the case, actually. If we look at this and look at the, you know, sort of this example clade, we can see a broad distribution of the different categories amongst various different clades, and there's no one like phylogenetic signal that can explain why a strain might be living in an ear or why it might be living in an eye, like, you know, necessarily. And like this example, you can see that there's a broad distribution, like even within a single clade. So this is actually a good problem for machine learning, because machine learning can tease out like underlying importance in the data set that may not be obvious by other methods. Okay, so just to talk a little bit about the genomics here. So each strain, after we've sequenced each strain, we then annotate, like whole genome sequence, we then annotate with a program called PROCA, which just goes through and attempts to identify all of the different gene sequences that exist in that strain. And so for us, what we're interested in is being able to sort of like cluster those genes together so that we can say, hey, these genes are the same and probably have the same function based on the fact that they're very similar in their nucleotide or amino acid content. So here we're saying they have to be 70% identical in amino acid content, and then we say they're all part of the same gene cluster. And so in this little table, you can see we have strains that are composed of these different presence and absences of various different gene clusters. And so the columns in here are the genes, you know, presence and absence, and the rows are the strains. And then finally, what we're really hoping to do is we're hoping that we can use machine learning to categorize each of these different strains into the different categories that they belong to. And we'll talk a little bit more maybe like about the sort of intuition about why we want to do that here in the next few slides. But this slide shows you sort of like a heat map, which is just exactly the data that I was talking about previously, except that it's been rotated. So now strains would be each column in this figure, and then the gene clusters would be the different rows. And you can see like the light blue cluster type on the edge of the heat map there, those indicate whether or not they are core. And core genes just mean whether or not they're present in every single strain, and if it's not core, then it's present in some subset of those strains. And initially what we're going to be doing is we're going to be looking at about the ones that are not core, because more than half of these genes are not core, right? And so maybe we can actually predict what area of the body or whether it was a carriage strain or not based on the presence of those different genes. So we have to do some filtering on this data. And the reason is because some of these genes are not actually very informative when it comes to trying to differentiate one strain from another. And so one category where they're not informative are core genes. right? Because every single strain has them, and so it doesn't give us any information when it comes to trying to differentiate one strain from the other. And the other category that doesn't really add very much is the genes which exist only in a single strain or only in one or two strains. Those don't actually give us a lot of differentiating power when we're talking about 1,600 different possible strains here. So we're like the graph that's on the left here, that is like the initial distribution of all the genes, and the one on the right is sort of like the middle distribution after we've filtered out the core and very rare genes. And that brings us down to about 794 different genes that we're going to try to use as gene features in our machine learning. Okay, so why would we want to do this, right? I mean, presumably, if someone is in a clinical environment and they isolate a strain of Haemophilus influenza, it's very likely that they already know where it is that they've isolated this strain from, right? And so, like, building a classifier to give an unknown genome and tell us where that strain is from is not necessarily like the most important reason to build a classifier like this. What's really important is that we want to be able to figure out what the gene content is that differentiates those different bugs from those different categories. And this can lead us to have different novel treatments for these different strains of bacteria that are associated with different places on the human body. And so I didn't actually talk about this very much previously, but all of these strains are non-typeable Haemophilus influenza. And in the past, people have used the polysaccharide coat on the outside of a Haemophilus influenza in order to create vaccines and boost immunity against those specific types because they have very obvious targets on the outside of the cell. And so since these are non-typeable Haemophilus influenza, it's harder to do that with. And so we're hoping that we can get some insight into these genes to be able to actually identify important virulence factors and new treatments and different gene annotations. And so one of the reasons that we have to worry about gene annotation is, so this is a word cloud of all of the different the words in the annotations, gene annotations that were assigned to MORAXELLA CATARRHALIS, which is another bacteria, one of the top 3 bacteria that's involved in middle ear infections, which our lab studies a lot. But essentially, like this graph is very similar if you're looking at a MORAXELLA or a strep pneumo or Haemophilus influenza. We get a very similar profile to this, and you'll notice that the #1 annotation to a gene that we've got is this hypothetical right in the middle there, this big guy. And so what that means is basically every time that we try to annotate one of these genomes, we do not know the function of most of the genes that exist in these genomes. I mean, it's hard to tease that out in a lab. It takes work and having someone dedicated to it. And so having these computational methods for us to be able to assign some kind of meaning to different genes is actually pretty important. And so this is kind of what we're referring to when we're talking about like the genomic dark matter. All right, so I'm gonna talk a little bit. I'm not gonna go into tons of detail about this, but just to give you guys some intuition about the algorithms that we're using, we'll do a quick history lesson about how you can use decision trees in order to classify different observations here, right? So the way that we're modeling this, if we're using a decision tree, is we can say, okay, let's pick some feature, and in our example, it's going to be a gene presence and absence. Let's pick a gene that like best splits our, all of our strains into 2 separate groups. And so the next step in the algorithm is, have we solved the problem? Have we perfectly split every, and assigned a classification to everything? And if we haven't, like we still have mixed classes in those strains that, after we've made that decision, then we go to the next level and we'll take another gene that best splits either the ones that had the previous gene or the ones that didn't have the previous gene, again, in the way that we can split them in the most even possible manner. And essentially, we just keep doing this until we reach our maximum depth of the tree that we've decided we want, or we have been able to classify everything, like every branch in that tree has a perfect classification, but only a single class. So this is a great idea, and it's a great first step. It's very simple and easy to understand, and also very fast to implement. Problem is, it's very sensitive to noise and variance, and you can very easily pick the wrong genes to split on initially, and just like increase the confusion of the classifier more than the accuracy itself. So the next like iteration of this is like, okay, well, you know, if one decision tree is good, then maybe if we use a bunch of them, we can get a better result. And that's basically the idea of random forests, is that essentially what we do is, instead of using the entire data set and only building a single tree out of that, to take subsets of our genes and build lots and lots of, you know, mini trees, you know, each one that can give a classification, you know, of a different genome, a different strain. And then what we're going to do is we're just going to like build a ton of them and take majority rule. And so this is sort of nice because it gives us properties that allow us to do things like decide what genes are actually the most important for classifying different strains, and ones that are you know, specifically important for particular classes and that sort of thing. It's a robust method. It was sort of king of a lot of machine learning competitions for years, but that was recently kind of overtaken by this next method, which is a boosting method. So, all right, you know, if more trees are good, then you know that increases our ability to classify correctly. Well, but are all trees that good? And so what the boosting methods do is they again are building a lot of different trees. So it's a very similar algorithm to the random forest. But this time what we're going to do is we're going to make decisions about the trees that were made, and ones that classify poorly in our training data set, we're actually not going to listen to them as much in the final classification of our strain. And so in this way, it actually can increase the accuracy because it reduces the poorly performing trees and the influence that they have in the final classification method. And of course, XGBOOST is sort of the king of this. Is like, the problem with that method is that it slowed the random forest down quite a bit. But with XGBOOST, there's a bunch of different optimizations that happened in the background, specifically doing things like parallelization and memory optimizations, which will make it very, very fast. In fact, even faster than traditional random forest implementations. So just a little bit about how I did this for people who are familiar with machine learning and want to, you know, check this. What I did is an 80-20 testing split. So that means that I'm training these models on 80% of the data, and I'm leaving out 20% of the data as to use as like a check. So like the models have never seen this data before, and I'm going to see how well training these models can classify these strains that they've never seen before. And so like it's also with five-fold cross-validation, which we repeated twice, and I implemented it in R with Carrot. Okay, so results. All right, how are we doing? So just using random forests in order to classify this, and the way that you read this little figure here, this little table, is the true class, right? So everything should be, you know, in that column, it should be whatever is labeled at the top of the column. So in the first column there, every strain is actually a carriage strain that we have labeled. And you can see every row on the side is what was predicted that strain, like those strains were. So you can see that 67% of our carriage strains were classified as carriage. And then like the next row down there in the carriage column, you can see like 5% of ear strains were classified as carriage, et cetera, et cetera. And so we can see here like that we were pretty good at classifying both carriage and lung strains as carriage and lung. And this is not too surprising to us because of that class imbalance reason that we were talking about before. You'll notice that along that top row there, most of the time when it's a different class than carriage, like the ear, eye, and invasive, when it's a different class than carriage and lung, most often the mistakes that we're making get drawn into that large class. And the reason for that is that because of this underlying imbalance, when there's a guess that has to be made, it more often guesses that it's part of one of these larger classes, because just by statistical reasoning, you would be more often correct if that was a true representation of the population in the world. But it's not. So this is a problem. And in a perfect world, on the diagonal, we would have 100%, because everything that was truly an ear would be truly classified as an ear, et cetera, et cetera. But we get about 55%, and that might not seem great, but it's way better than if we were just randomly assigning these based on their class frequency, which would be around 32%. So there's definitely information in there that's allowing us to get a better classification than random. All right, so what happens if we use XGBOOST? So we get better, right? So our class accuracy goes up to about 60%, a little over 60% there. We're still having trouble with those imbalanced classes, right? So again, you can see more often than not, if there's a mistake being made, it's assigning the mistaken genome to one of the large classes, either the lung or the carriage class, and less often to the ear, eye, and invasive class. Okay, so that's good. So can we do a little bit better? All right, so there's actually some different parameters that we can modify for these different algorithms. Random Forest has a couple of different ones. Mainly, it's just the depth of the tree, like how many genes you allow to be in a tree, or like the gene layers in a tree, and also like just the total number of trees that are created from randomly sampling from your data set. XGBOOST has a bunch more different parameters you can modify. I kind of focused on like the learning rate, so how like the amount of modification that happens whenever XGBOOST is trying to make some decision about how to weight things. How deep the number of layers that you want in your tree, and your optimization rounds. Those are the ones that I sort of focused on here. So we did a little optimization parameter search space, looking at a variety of different numbers of rounds, changing the learning rate. It defaults to 0.3. But I wanted to kind of look at, what happened if we let it learn a little slower. And so like when it saw something mistaken, it didn't like buy into that so strongly to begin with, allowing it to sort of like more slowly decide what were important variables. And then like looking at what the tree depth was. So like the number of different genes that we would include when we're building our decision trees. And so you can see from here, like actually it looks like, like the learning rate at 0.3, this middle graph here, would be way better if we were much lower than that. In fact, like I might even, continue to go a little bit lower. And one of the problems with doing this parameter search space is that it's hard to change different parameters and then make decisions about them without fixing other ones. And so this is something I'm going to talk a little bit about what we're going to do in the future, but at least it certainly looks like reducing the learning rate to be a little slower was advantageous. Also increasing the number of tree levels appears to have like an increase. Once we get up to 20, basically there's some diminishing returns, but certainly more than 6, right? More than the default. And in the first graph there, like it really doesn't seem to change very much how many rounds of optimizations we do. In fact, it almost looks like we might be doing worse if we do too many of them. So yeah, so I changed it, and do we do any better? And the answer is yeah, kind of. Not tons better. Basically our carriage gets a little better. And there's some movements in the class distribution, class predictions, but doing tons better isn't what I found here. And part of that I think is due to the way that we did the search, or I did the search in that space, and I think that we can do a better job with that. But the whole reason we did this, right, is to identify some important genes, and we were able to do that. So after building these models, we were able to identify like the top 20 most influential genes. And what's really interesting about this is, again, we get some things that we sort of maybe expect to see there. So like, for example, there's a beta-lactamase, which is protection against antibiotics. But there's a bunch of hypothetical proteins and unknown proteins, proteins of unknown function. And so that, is sort of exactly what we want to see. We want to see this unknown biology and ascribe some meaning to that. Okay, so the next strategy that we can use, and I'm going to have to kind of go through this pretty quickly, but we're leaving out a ton of information, right? Like we don't use the core genes at all. And so core genes are ones that exist in all of the different strains. And maybe there's some information in that, like the genotype of them themselves, that would also be very important for us to know about. But the problem is with using core genes is that it's really difficult to compare very different genes. Normally what we do is we look at them in alignment space. But in order to get a good alignment, they have to share a bunch of homology. And so you can't just compare one gene that's vastly different to another gene. So for example here, in this top part here, we've got two very different genes. And the dark blue lines are just places where they have exactly the same amino acids. But basically, this just means that they're from the same alphabet. They don't actually have a lot of relation. So we need some way of comparing very different genes with very different lengths and very different identities to each other in some functional space. And so you can see, like in this bottom here, that we've got these two very different genes. If we could translate that into a vector of numbers that meant something in their functional space, then we would be able to devise some function of being able to compare those two together. And so it just so happens that Dr. Dampier, who is also part of this conference and has a talk, I think, right before mine, also about deep learning, he created a method for us of transforming these proteins into vector space so that we could compare any coupling of proteins that we wanted. And I won't talk very much about that at all. I encourage you to go check out his talk and see, to learn some more about the underpinnings of that process. But what we find is if we take 200 of the core genes and then we use their vectors to create a two-dimensional plot like this. So this is a t-SNE plot, which is just like a PCA plot or an NMDS or what have you. It's taking a high-dimensional thing and collapsing it down into 2 dimensions. You can see very good grouping of the different genes next to each other, and I've labeled them with our own predictions of what cluster they belong to. And so what this means is that Dr. Dampier's method is capturing the underlying biology. So even though it's going to a vector of numbers and it's hard to say like what each individual number means, it is capturing something in functional space that groups things that are similar to each other. So I use those vectors in order to run a couple new, or another iteration of the XGBOOST algorithm, this time using the summary of those vectors. So like the sum, the mean, min, and max of them. And I said, can we predict the results from those as features instead of using just gene presence or absence? And we actually do better. We do better than we were able to do with the distributed genes. So we're up to around 61%. Again, way better than just guessing. And yeah, I mean, like generally speaking, we actually do much better, especially in the I class, of being able to differentiate those apart from the others. So there's definitely a lot of information here that we can probably use. And again, like the whole reason for doing this is identifying important genes, and we can do that. You can see here that we can identify a group 1619. Many of those summary metrics pop up in these most important differentiating gene vectors, right? And so like that's a really important group that we'd like to take a look at. As well as some of these other ones that I've got colored arrows by. And this is very green data, so I can't really tell you very much about them. But you know, if we reduce the number of classes, like we get rid of some of those low, fewer like classes with fewer genomes in them, then we do better. We can get up to like 71%, but that's a little bit of cheating. And what we'd really like to do is do some other strategy of taking care of those imbalanced classes. And so in the future, obviously, like there's a bunch of different things that we can do here. Search the parameter space for optimal parameters for the different algorithms, do some kind of fancy imbalance, of classes, handle that a little better. Do some feature selection before, like get rid of features that are just not very informative and actually just noise. Examining our misclassified genomes and trying to make some decisions about that. Maybe making some decisions about the phylogenetic signal and the ones that are clustered together, and maybe we want to reduce those. Also look at classifying our other and unknown genomes and just see what we get. And then, of course, like combine those two approaches. Maybe we can make the granddaddy of all classifiers and really get a much better prediction if we use all of the data instead of just some of the data. Thank you very much. That's the end of my talk. I think there's going to be the chance to send me questions via email. Thank you very much to the organizers for having me. And, obviously, like Josh Mell, Jocelyn Hammond, and Rachel Ehrlich both contributed a massive amount to this work in their curation and bioinformatics. So thank you very much, and that's the end.